Hey folks, can you guys let me know in the chat if you can hear me? Great. Give people a second or two to trickle in. I know that unfortunately we had some issues getting up and going, but hopefully now people can see the meeting and are gonna trickle in. I want to thank Holly and Feather and the rest of the organizers for inviting me here to talk to you guys about CellProfiler. Wish we could all be in sunny, pleasant California, as opposed to right now I'm in snowy Boston. But it's nice to meet you all and hopefully answer your questions about CellProfiler. I'm going to go through and talk a little bit, give a PowerPoint presentation, and then I'm going to demonstrate the software. I'll be trying to keep an eye on the chat as well, so please feel free to put questions there, and I will do my best to answer them. My name is Beth Cimimini. I work at the imaging platform, which is also known as Ann Carpenter's lab at the Broad Institute. I'm a CZI imaging scientist, and I've been a member of the CellProfiler team for about 5 years now, and I want to talk to you guys a little bit about sort of why you might use CellProfiler, why we think CellProfiler has a place in the wonderful, huge, chaotic, fantastic open source image processing ecosystem, and then give you guys a little bit of a demonstration and answer any questions you have. All right. And again, I will try to keep an eye on the chat if my screen setup will allow. Right, let's see if I can get both screens up at once. So we're passionate at the imaging platform about doing image analysis because hopefully I don't have to convince this audience too much. The images can contain a tremendous amount of information. You can multiplex images, and even if you don't, every time you... Oh, hi. Someone else was talking. Anyway, I'm gonna go for it. Every time you snap a 2000 by 2000 pixel camera, which is pretty standard most days, you get 4 million pixels worth of data. And this is quantitative data that you can mine. And you really can't get that amount of information richness in really almost any other modality. Some of the sort of like single cell RNA-seq stuff is starting to come along, but to be able to quickly look at thousands of cells and millions of pixels all very quickly, we still think imaging sort of takes the cake. This can be sort of a controversial thing, though, because in general, image analysis did not used to be technical. If you ever do have a chance to see the beautiful drawings that Ramoni Cajal did, they went on tour around the US, I know, a few years ago. It used to be image analysis was drawing what you saw and hoping that it was representative. And a lot of people still conceive of imaging that way. I'm personally very glad I didn't work in that time because I would never be published because all of my drawings would be stick figures. But given the advent of the digital camera now, and again, like looking at some of the FLIM data we've seen today, you know, certainly incredibly quantitative information can now be pulled from images. With the digital camera, images are just arrays. So on the left and on the right here, we have a digital image, and we have the underlying array that it's built from. This is a very simple example that I made quickly in a Jupyter notebook. Here's a more complicated one. And even though this is much more a picture to our eyes, it's still ultimately just an array of numbers. And we can examine that array. And so then we can do quantitative processing on that array. And so this just simply sort of brings home the idea that visual appearance is a thing that can be quantified. And we want you to do quantification by an image analysis software when you can, even for something sort of as simple as localization that it looks like you can see by eye. And there's a few different reasons for that. It's objective, it's quantitative. You can look at heterogeneity, which is really hard to do when you're sort of counting things by hand. It's faster and less tedious. You guys have a tremendous amount of training, and you should be spending your time either in the lab doing something really amazing or out of the lab doing something really amazing, and not spending time necessarily sort of counting cells by eye or tracing things. And I want to come back to the next last bullet point, which is also that Quantitative image analysis allows you to detect changes that you can't see by eye. Now, the wonderful thing about the human brain is it's really fast at sort of giving you an idea of what's going on in a scene. The bad thing about the human brain is it takes shortcuts to get there. When I first put up this picture a few seconds ago, you probably originally thought it was a color picture. It's not. This is a grayscale image with some colored lines drawn over the top of it. Our brains are trying to give us the idea of what's going on around us as fast as they can, and it takes shortcuts to get there. And it gives you the basic idea while not necessarily accurately representing the details. So even if you have a phenotype where you think that you can see it very well, and you don't have enough, that you're not interested in heterogeneity, you don't think you have enough images that it's worth building an image analysis pipeline, I would try to convince you that it is, because your brain lies to you a lot. And there are many amazing open source image analysis tools. Ours is one in as I said, a vast, wonderful ecosystem. Almost everyone here, I'm guessing, has used ImageJ or Fiji. I'd be curious if anyone hasn't. But this is often the first image analysis software you learn to use as soon as you've sat down at the microscope. And I use this every day. We work closely with Kevin's team at Madison. It's very fast, and with the advent of Fiji, the plugin ecosystem means that there are tons of plugins that are available to do exactly what you want to do, exactly how you want to do it. The downside with Fiji is that unless you're working with one of those plugins, and unless it's a plugin built for batching, if you have an analysis that you want to do over and over and over again, you have to be pretty comfortable scripting or writing macros. Now, ImageJ has a great macro language and a scripting interface so that you can do this, but not everybody wants to, and not everyone should have to in order to do their image analysis. Another great tool is Elastic. Elastic is wonderful for when you have an image where you can see the difference here, but you don't necessarily have a fluorescent stain or some sort of stain that stains exactly the part of the cell or the organism that you're interested in. Elastic allows you to use the principles of machine learning to essentially draw on the image in a way that, for those of you who are old enough to remember, like me, Microsoft Paint allows you to do. And that will train a machine learning classifier underneath, which allows you to distinguish many different parts of an image. And so Elastic is a great tool that we also use quite a lot in our group. But the disadvantage is that it's great for telling you where structures are in an image, but it doesn't often tell you a lot about their properties. So this is where we think CellProfiler fits into this ecosystem, and also its companion to CellProfiler Analyst. What CellProfiler is designed to do is to allow you to make an image analysis workflow and to apply it to every image you have, whether every image you have is 1 or 10 million. And 10 million is not an exaggeration. I've done 10 million image sets in CellProfiler. It is designed to find the things you care about in an image and measure them. Once you've done that, and once you've created a spreadsheet or a database with all of the measurements that you care about, CellProfiler Analyst is designed to allow you to actually use that to answer a question. I mostly throw this slide up there just to show our amazing software engineering team who've worked on the software, from Ann Carpenter, my current boss, and Ray Jones, who wrote the first version back in 2004, all the way through to today, where we have David Sterling and Alice Lucas are our newest members of the team. And what we're also really proud of is that the software is free and open source, and that it's been thought of by the community as flexible and usable. CellProfiler has a lot of options. One might even say too many options, and we'll talk about the different kinds of things CellProfiler can do, but it's designed to be very flexible to allow you to customize the image analysis you want for the biology you want to find. There are several, you can't tell from this image, but also what's, I think, a little special about CellProfiler is about half of the people that I've shown here are wet lab trained biologists, and about half of them are professional software engineers. We've basically always had at least one of each working on the tool, which means we have the benefit of trained software engineers who actually know what they're doing and help us write great code, and wet lab biologists like myself who understand really how biologists think, what are the kinds of assays biologists want to do, and help figure out how we can bridge the divide between those two communities, which often have very different sets of language and ways that we talk about problems. So as I said, CellProfiler is designed to take an image, find objects in it if you have objects that you care about, and then export data for further analysis. Our lab philosophy is measure everything and ask questions later. We want to make it easy for you to do lots and lots of measurements in CellProfiler. And hopefully during the course of this talk, and the demonstration, I'm going to convince you that actually generating lots of measurements is a really valuable thing. And the goal is to make that easy for you to do, whether you're an expert software engineer or whether you've never opened the command line in your entire life. Almost always in biological image analysis, there are two main steps to accomplishing finding out the biological answer that you care about. The first is to find something in an image, and here we have just two examples of what some things could be. We have some human nuclei and we have some yeast red-white colonies, and then to measure something about them. The first part is the hardest part. Once you've found something, it's pretty easy to look at lots of its properties, but the uniqueness comes in figuring out how to find things. And so we provide a number of examples on our website because our goal here is that we don't want you to actually have to figure out how to do this by hand. There are a lot of different kinds of biological images you might be interested in looking at. And we want to help you get started by sort of giving you some examples that you can start from so that you can change the pipeline to fit yours.
Hello. I see you made your way to the testing room. I didn't think to send it to you, but you found your way here anyway.
Hold on a second. I have apparently a different window open. Let me
did I send
technical errors here.
Or you just happened to run into it?
Sorry, guys. My next slide was about to show you the CellProfiler interface, and now I can show that to you live. This is the CellProfiler interface. This is what it looks like when you open the program. Our current release version is 4.07. 4.1 is coming out sometime in the next couple of days. And it's divided into essentially two halves. On the left half, we have what we call the pipeline panel. And the pipeline panel is essentially where you specify all of the steps that your image analysis project is going to do. That might be something like smoothing an image or subtracting its background. It might be finding objects. It might be measuring objects. When I add any modules to my pipeline panel, what I see is on the right, each module, when I click on it, gives me all of the individual parameters that I can tweak. So I have a list on the left of all of the things I'm going to do to each and every image when I load it, and it will always happen the same way for every image. And on the right, I can configure the details of what that looks like. The first step in using CellProfiler is to use the input modules, which are the top four modules here: image, metadata, names, and types and groups, to use some combination of some or all of them to create what we call an image set, which is however many channels you're looking at a period of time. CellProfiler can be used whether you have one RGB histology stained image, whether you have images where you have many different channels, many different time points, many different z-planes. And you just need to tell CellProfiler what you're doing because it doesn't magically know how your experiment is configured. You then go through and add different kinds of things to do, such as image, object processing, or measurement, to go through those steps I've outlined a couple times now. And then at the end, to add file processing modules to write out all of your images and measurements to a disk or a database. I said before that one of the problems with CellProfiler is it can be a little bit overwhelming. We have about 70 different modules that represent all of the different things that you can do within CellProfiler. That notably doesn't even include the plugins that other people can write that aren't part of the main CellProfiler program. Because of that, we do have everything organized into categories. So you can go through and say, well, I know I want to do something to my image. You'll find that under image processing. If I know I want to measure something now, I find that in the measurement category. You can also search the modules for keywords. You can type in size and you'll get, for example, the measure object size and shape module. And that will come up, if it's in the module title, it will come up there. Otherwise, you can search the help text for every module. When you look at images in CellProfiler, they look like this. This is just an example, an image of some nuclei. You can zoom in, you can pan, go forward and back through the different views you've made. You can save out the view you're currently looking at, or you can hit the home button to go back to the start. When you look at the bottom, you'll see the XY information of where your cursor is as you're hovering over the image, and also the intensity under that. So if you want to say, all right, I eventually want to threshold this image so all of the nuclei are thresholded out, how bright are the nuclei? You can hover your pixel over and find out. This intensity information may look a little bit different than other tools you've used. Some softwares scale intensity information 0 to 1. Some of them do it sort of 0 to bit depth, so 0 to 255 or 4095 or 6535 typically. We rescale everything 0 to 1. So exactly be identical to what you're used to looking at in, say, ImageJ, but it's the same information, it's just a decimal instead of a whole number. You can also do things like measure the length of objects. So if you go to the measure length tool option and you click and drag across a nucleus or whatever object you care about in the image, you'll get an idea of what the length is for as long as you hold down the mouse. I've said a couple times now that the value in CellProfiler is that we can find objects, we can do a lot of different things, but then we can measure them. And I want to now explain to you why we think that's pretty important. And one of them is just that there are a lot of properties of biological objects that are interesting. And what CellProfiler does is it makes it pretty easy for you to get all of those properties in adding just these dozen or so measurement modules that we have. Most of these metrics are not unique. Most of these metrics can be found in lots of tools. In fact, when I'm demonstrating now some of the metrics you might want to look at, I'm going to actually show you some things from Fiji. So CellProfiler isn't unique in that, but what we think it adds value is it helps make it easy to look at lots of them all at once. If you add the measure object size and shape module, for instance, you're not just going to get the area, but you're going to also get the perimeter measurements like eccentricity, which basically says, is your object more like a circle or more like a line? How solid is your object? Are there holes in it? How is it oriented? You'll even get some pretty fancy measurements like Zernike polynomials, which are essentially, and you don't have to worry about the math here, shape-based descriptors that don't really that's in the human brain, but essentially are how much is an object sort of shaped left to right versus up and down versus in a circle or not. And so this is what they look like as a 3D representation. And you might say, if I don't understand what this means, why would I want to measure it? And it's because it can give you a pretty accurate description of an object shape, whether or not each individual measurement means anything to you. This is one of the original papers that proposed this for image analysis as a metric. They measured lots of different biological images under a microscope, and then they said, all right, if I take some of these Zernike polynomials, how accurately can I reconstruct that shape just from a few mathematical measurements? And it's not a perfect representation, but it's also not a bad one either. It gives you a pretty strong idea of how an object is shaped with just a very small number of mathematical descriptors. And so this might, measuring Zernike polynomials, whether or not you intuitively understand what they mean, would still actually help you find out things about the object shape that you could see if they're different in, say, control versus treated. When you add the measure object intensity module, you will get not just the mean intensity, but also the median, the standard deviation, the sum. And if you have multiple channels in your experiment, you can take any channel and measure in any object. So if I have DAPI and GFP, and I find my nuclei in DAPI, I can measure their GFP intensity. Once you've found an object in any channel, you can measure it in every channel. You can also measure the distribution of that intensity. Is it all evenly distributed throughout my whole object? Is it more on the right versus the left? Is it more on the inside versus the outside? All of that might be really important for describing your biological phenotype. You can measure the texture, which are essentially how smooth versus bumpy are my objects, and we include different modules to measure each of those. And again, the underlying math here isn't really that important, but essentially it says if I'm at a point in an image and I go to the next point over, is the intensity pretty similar or pretty different? And that helps you to understand, again, is your intensity smooth across the board, or are there lots of bumps or striations that might tell you something about your biology? Colocalization between stains is, of course, a really important one. This is not the CellProfiler colocalization visualization, but the one from Fiji. But just to show you, again, the sorts of information you can measure, and we measure several different measurements of colocalization, such as Pearson and Manders and Spearman and a bunch of others. Essentially all the ones you would find in the Fiji colocalization plugins. And once you're done, you can write all of this out. You can write it to either a spreadsheet, so you can open that up in something like Excel, or you can write it out to a database. You can put it into an SQLite database, which is just a small container file, or if you're working on a cluster and you have thousands of images, you can write to whatever MySQL host your university or company provides. You can also write out images, and this can be really critical when you're working in doing image analysis, especially once you have more than a few images. There's nothing more tragic than thinking that you have found a really cool phenotype because a measurement changes between your untreated and your treated samples, whether those are genetic treatments or compounds or mutations or what. And then you realize it was just that your segmentation was nice in one group and not in the other group. So being able to look at the measurements or look at the images of what actually you found are really important, and CellProfiler provides you an easy way to do that. The final thing that it allows you to do is also just to save the analysis you've done itself. You can save the pipeline, which is just the list of things that are on that left half of the program, and you can send that to your lab mate. You can submit it as supplemental material in a paper, and everyone can make the same exact analysis you did. We have an even easier thing to use for when you're working yourself on your own computer called a project file, which also saves in the image location information, so that if I am working on something and then I go to do something else and I want to come back to it, I can just load the file and all of my images are already loaded in. So I'd like to go into a quick CellProfiler demo here and show you just a couple different kinds of pipelines we can put together. When you first open CellProfiler, you're gonna get what we call the welcome screen. And it's gonna have a few different links, such as to the manual or to link to our tutorials page or to our forum. If I hit try example, it's gonna load up an example pipeline that I can use to demonstrate or to try it out on your own. What you hopefully will notice are a few things. One of them is at the top of each module is a place to take notes. So you can leave notes to yourself or to whoever uses your pipeline after you about why you may have set certain things the way you have. You can say that, for example, that this module is a module that is designed to find nuclei from the DAPI image. And that in certain circumstances, if you have a thresholding option that does 3-class thresholding, it does better than 2-class thresholding. If you don't understand what that means, that's okay, because what we are most proud of in CellProfiler, at least what I am most proud of, is that we have a lot of different modules, and many of them are configurable in a lot of different ways. This is a pretty extreme one. Most of the other modules are much simpler. This is a module just for cropping something into, say, a rectangle. Every single setting of every single module has a help button next to it. So you can hit this question mark button, And you can choose, and it will tell you exactly what that setting does. If I hit the question mark button that's in the pipeline panel, it gives me the information about the whole module and what that module does and what each one of its settings does. And so there's a lot of things that you can do in CellProfiler. Having all of that power can make it a little bit difficult to understand what each thing is doing. And we don't want you to ever be lost. So by using all of these individual help settings, it'll tell you how to move forward. And if you, once you've figured it out, you can leave a note to yourself on what you want to do for later. CellProfiler works in two different modes. There's the test mode and the analysis mode. Test mode is when you go through and you take a look at all of the different settings and what they're doing. This is where you're going to spend 90% of the time that you're sitting at your computer using CellProfiler. Once you're done, you hit analyze, and however many images you've loaded, whether that's 3 or whether that's 3 million, will run through the program one at a time through the set steps that you've outlined here. So what do I actually have here? Well, I have 9 different images that are loaded. I have a D, an F, and an R at each of 3 different positions. And so these work out to be 3 different channels in my experiment: a DAPI channel, a FITSI channel, and a rhodamine channel. CellProfiler doesn't know that automatically, and so we can use the names and types module to teach CellProfiler what's going on in our experiment. So here I've said that have d.tiff in the name are blue, the ones that have f.tiff in the name are green, and the ones that have r.tiff are red. And you can put in whatever names you want. If you have grayscale or color images or even object masks from something like Stardust, you can load those in too. There's a lot of different things you can do here. I'm going to skip metadata and groups because those are modules that you only use under special circumstances. When I hit start test mode, I'm going to get a few extra icons over here. I already had a check mark icon and an eye icon. The check mark icon essentially says, should this module be run, yes or no? So if I turn it off, CellProfiler thinks I'm not using it anymore. And so that's an easy way to sort of play around with your analysis, see what's going on there, and then if you don't like something but you're not sure whether it's useful or not, you can uncheck it and you can leave it and decide whether or not you want it later. The eye being open tells you whether or not CellProfiler shows you what it's doing. So if I run this first module, it shows me that it's cropping this large image down to a small area. If I run it again with that eye closed, nothing happens because I've told it not to show me what it's doing. The green arrow points at what's going to run next. The gray arrow points at everything that's already run. And these are pause buttons. So what I can do is when I want to execute modules, I can say step, and it will go and just do one more. Or if I hit run, it will go until it hits a pause button. So if I hit run now, it'll do a third cropping module, and it will do the identify primary objects module, which is typically the module you would use to find objects. So here's what that looks like. It shows me my input image. It shows me the outlines of all of the things that it found. And it shows me then a color view of that. You'll notice there are different colors here. Things that are circled in green are things that it recognized are good objects. Things that are in orange are things that are touching the edge. And so right now, CellProfiler is set to throw those away. I can very easily set that to no and rerun. And now those objects are green and they're included. And I can also, if I have any objects that are the wrong size here, I can see that because those are outlined in pink. And those objects are not included up here in my color mask. And so you can have a lot of control over whether you take a large variety of objects or whether you want to really focus in on a very certain kind of thing in your image. Once I have my nuclei here in my DAPI channel, I can grow those out into cells. Typically, we recommend that you do that. because nuclei are often a lot easier to find, and if you're working in a mononuclear cell line, it is a lot easier to sort of start from a nucleus and build a cell around it, but you don't have to. You can find cells. I see a question that says, can I talk about the underlying segmentation models? So right now, most things in CellProfiler and everything that's in the main program for CellProfiler is what we call classical segmentation, which is typically a thresholding followed by some sort of a watershed. We don't have any network-based or other segmentation models or object identification models in the main program of CellProfiler, but you can actually download some of those as plugins if you have CellProfiler installed in Python on your machine. And so I've actually written some plugins where you can create a deep learning network in some other program, and you can actually use that profiler as a plugin. But all of the things that are in the main program are conventional segmentation where you do a thresholding and then a watershed. And that's simply because at this point, deep learning based models, while we think they're really powerful and we think 5 years from now they might essentially be everything that's in CellProfiler, they're really difficult to package right now. And we want this to work on everybody's computer, whether they have access to a top of the line GPU or whether more likely they're using the 8-year-old lab computer that's in the back next to the microscope. Once I've found my objects here, I have some measurements. I have some visualization models that I can use, such as this gray to color, which is going to combine my cropped area into an output thing that I can look at later. So when I want to say, all right, what is the count of objects and sort of do a human head check on it, this image can be saved out. And you can see here, I've created a large number of measurements in just a few seconds across my whole image. I've measured things that are in the nucleus, in the cell, and in the cytoplasm in a number of channels and in a number of ways. Most of the time you spend in CellProfiler will be in adjusting your object finding. That's the really hard part. So we do also offer something called the workspace viewer, where I can load up all of my different images. And if you have, for example, a really tricky cell, you're having a hard time, helps if I load either all cropped ones or all original ones. I can overlay where all of my objects are, say my nuclei. And if I'm having a really hard time looking at, say, just a couple nuclei, I don't find this color that visible. I'm gonna make it teal. I can zoom in on a particular area. I can play with my segmentation parameters, such as I can say, instead of doing what we call a minimum cross-entropy threshold, I'll try an Otsu threshold. And then I can take a look to see in that area how it affects the segmentation that I have. And in that case, it's doing a really bad job. So we probably want to go back to what we had. But it allows me to look at a small area and see what's going on and to turn individual channels off and on. And if I have more than one object, I can also look at more than one object at a time. And so this can be really helpful as you're building your pipeline. Eventually, you want to run your pipeline. And so you're going to exit test mode. I can go to Window, hide all windows, and it's going to close all of these eyes and close all of these windows. I can say that I want to output this to my desktop in my AIM 2021 folder, and I can go ahead and hit Analyze. And this is going to analyze all of the image sets that I have. In this case, it's 3. If I had more than 3, it would analyze as many as I had loaded in. It's running 3 CPUs at a time because there are only 3 images, but it's going to default to running as many CPUs at a time as you have on your computer. You can tell it to use less if you want, but that means if you have 1,000 images to run and you have a 16 CPU machine, for example, it's not going to run 1,000 images on one CPU. Each one is going to run 60, and so it's going to complete a lot faster. And this should only take a minute or so to run. I can pull up another my other CellProfiler window, which is now running away from me. Okay, this is rude. All right, So I can also, finally, open up an example pipeline. So these are all of the examples that we have on our website. I can pull up what we call the speckle example pipeline. And I won't go through this one in as much detail because I think you guys get the sort of basic idea, but to sort of show you the variety of kinds of analyses you can do, here I have a nuclear pipeline or a nuclear image. And I have a gamma H2AX antibody, which is finding DNA damage speckles. And if I go into test mode, I can do, you can see a few of the image processing modules, such as I can find my nuclei. I can take my speckle image and I can remove all of the background from it. Let me zoom in so that's a little easier to see. And let me also adjust the contrast here so it's a little easier to see. So there's a lot of haziness that's from out of focus light in this image. And I can apply what we call the enhance or suppress features module to pull out only the speckles and ignore all of that background staining. Since I know that my foci are going to be in my nucleus, I can also use the mask image module to say I only want to look in the H2AX channel within nuclei. And so this is where your biological training actually becomes important, is that you can go through and you can say, all right, I know where the object I care about is, and you can customize the pipeline to see, you know, do I only care about speckles in the nucleus or not? We leave the decision up to you because you know what you want to look at more than we do. I can then identify these small speckles that I found. So that looks to me like it's doing a pretty reasonable job of finding the speckles. There's some dim stuff here that I'm not quite sure is real or not, but again, you can customize the pipeline to be as strict or as permissive as you want. I can then do a bunch of measurements on my speckles and on my nuclei. And I can relate the speckles back to the nuclei. So I can say, all right, all of the speckles that you found that were inside this bigger object, make sure that you know that those belong together so that when you go to look at your measurements later, you can say, all right, how many speckles of H2AX did I find in each nucleus? So those are just a couple examples of those sort of many different things that you can do with CellProfiler. There are pipelines for looking at C. elegans. There are pipelines that are designed to look at histology images. Someone asked about 3D volumes. You can look at 3D as an individual series of stacks if you want, but in the names and types module, I can specify that a particular image set is 3D. And then I can look at it as a whole volume at once, not just one Z-plane at a time. Great question. All right. So in the last few minutes, I want to talk about measurements. And again, I've talked a lot about that measurements are really important. And I just want to talk a little bit more about some of the reasons that we really try and make measurement handling so easy. And this is actually, again, sort of not the way a lot of biological image analysis is done. This paper's a few years old now, so I'd be kind of curious to see if this is still the case. But even in papers that describe themselves as doing high content microscopy, most of them only use one or two features of the cell. Now, I've told you in this talk that you might have 4 million pixels in an image and you might have multiple channels. So we feel like using 1 or 2 measurements at a time, while it can be really valuable, and I'll show you an example of a project that we did using a single measurement, there's a lot of phenotypes that we think are out there that people haven't been taking full advantage of and where they're sort of leaving data on the table. And so we want to allow people who who are using CellProfiler to get access to all of that data, even if they're not sure ahead of time that they need it. These numbers change a little bit when you look at people who were specifically saying they're using CellProfiler, but they're pretty similar across the board. And again, you can do really good biology with only one measurement. A tremendous number of people have. So this is data from our lab from a while ago now. We were working with John Crispino's group at Northwestern, trying to screen for drugs that cause cancer cells to not necessarily die, but to start endo-reduplicating and sort of duplicating their DNA without dividing and just get bigger and bigger, because those cells were no longer dangerous to the patient in AMKL. And you can figure that out with a single measurement, which is the sum of the intensity of the DNA or Hooks channel inside each nucleus. It's actually really easy biology to do, and there's now clinical trials underway based on having only a single measurement. Some phenotypes you can get away with too. So if I want to know cell cycle with pretty high accuracy, for example, if I have a DNA marker and I have a mitosis marker, I can look at essentially where I am on that XY plane of DNA content and phospho H3 and see whether or not I'm in anaphase, telophase, G1, G2, and again, with pretty high accuracy. But there are an awful lot of phenotypes that really don't lend themselves to only one or two measurements. All of these are phenotypes that can be distinguished by imaging and by imaging measurements, but I personally couldn't tell you what is the name of the measurement that does it for each one, for any one of these phenotypes. And to distinguish all of these phenotypes, you'd certainly need many measurements. And so this is why we're in CellProfiler, we try to make it really easy, because a lot of times the biology you care about isn't so easy to define. And so in our lab, we really like the idea of what we call the morphological profile, where we're sort of used to thinking about in an individual cell, taking a look at seeing which genes are up and down. Again, like in an RNA-seq experiment, we can see which genes are overexpressed or underexpressed or not expressed at all. But you can do the same thing with image-based measurements. You can see if the intensity or the texture or those Zernike features that I mentioned, which again make no intuitable sense to most human brains if you're not used to thinking in that sort of mathematical space. They don't make any sense to my non-mathematical brain, but we can start to build up big profiles, and then we can start to group cells and see if there are, for example, changes in groups depending on whether my cells have a mutation or whether I've treated them with a drug or whether they come from a patient versus not a patient. And there's a tremendous amount of work that our group does that I'm not going to talk about related to that, maybe next year's AIM. But so we also provide a tool called CellProfiler Analyst, because CellProfiler essentially just takes you from images to spreadsheets or databases. And as much as I love spreadsheets and databases, it's not actually the answer to a question. And so we've provided this tool called CellProfiler Analyst so that if you have spreadsheets or databases based on images, whether or not you did that analysis in CellProfiler or not, so they're designed to work together, you can then explore your data and do things like machine learning to actually try and find your phenotypes based on what you've measured. So I said our lab motto is measure everything and ask questions later. CellProfiler is the measure everything, and CellProfiler Analyst is the ask questions later. This is also the name of our blog. And I'm really not going to talk too much about CellProfiler Analyst other than to sort of show you it allows you to do scatter plots and histograms of the things that you've measured in CellProfiler. If you're looking at stuff that's in a multi-well plate, you can look at how each object measurement or image measurement changes in each well of a multi-well plate. So if you want to see if, say, cell count is down in certain wells you've treated with certain drugs, CellProfiler Analyst makes it easy to look at that. You can also do machine learning, and I've said that term a few times now, but I haven't really explained what it is. Probably some of you know what it is, and probably some of you don't. So for those of you who don't know, the idea behind machine learning is just that we're going to allow the machine to figure out what is important. And so if I, as the biologist, care about these sort of yeast-shaped cells, I can use CellProfiler Analyst or any other tool that's designed to do machine learning to separate my individual cells into groups. Now you'll notice next to each drawing of a cell, there are these different sort of thick and thin gray lines. So what that's designed to represent are all of the different measurements. I showed you that we can collect a whole lot of measurements for each cell, and some will be up and some will be down in any given cell. And so that's represented here by line thickness. And by having a lot of measurements, we can actually allow the computer to learn some rules about, are certain measurements predictive? And again, it means that you don't have to know ahead of time what are the mathematical measurements that describe the phenotype you care about. You just have to be able to describe it and to show it and to use it to drag and drop cells into bins. So CellProfiler analysts will do the hard work of then actually figuring out the math behind it that allows you to then sort of apply this. But all you have to do is then go through an iterative process, say, all right, have you figured out E-shaped cells yet? And then it'll show you some it thinks they are, and then you're either say, yes, those are the right cells, or no, they're not. And just the last sort of slides I want to talk about is a group that was not our lab, but was using CellProfiler to sort of do this sort of approach. They were using this in patients who had different blood cancers. And unfortunately for all of these patients, they'd had blood cancer that had come back at least once after treatment. And so because these are blood cancers, it's really easy to take blood cells from a patient and to get a lot of cancer cells. And so what they did is they took cancer cells from all of these patients and they put them into multi-well plates, and they tested a bunch of different clinically approved drugs. And they created a machine learning based classifier using CellProfiler features to figure out which cells were alive or dead. And because they knew something about each of these patients' cancers, they could figure out, in general, were all of the cells dead, cancerous and non-cancerous, or were cancer cells dead and normal cells were alive, or vice versa. And so they could generate a metric of how specific was this drug treating their cancer, and do it only based on machine learning measurements. About half of the patients then, their physicians still thought that they knew what drug would be best to treat their cancer that had come back. And so for those 13 patients, the physician chose the treatment. And for the other 17, in general, there was no obvious good next drug to give them. And so they allowed the machine learning to pick which drug to give the patients. And so this is before this, for the cancer that the patients had before they came into this, there's Kaplan-Meier curve of how long their cancer went without coming back. Everybody's cancer came back or they wouldn't have been in this study. And if you look at the people who got drugs that were picked by machine learning and by image analysis, what you see is that about half of them a year out still don't have, their cancer still hasn't come back. And in general, they do much better than they had on their previous drug. If you look at all of the patients, including the gray dots, which were the drugs that were picked by doctors, and the blue dots that were drugs that were picked by the machine learning, you see as in general, the blue dots tend to do a lot better, and they tend to have either a partial response or a complete remission, because they were able to tell just from growing their cells in a dish which drug did best. And so this is precision medicine, just based on image analysis and creating a machine learning classifier for cells that were alive or dead. So this isn't a silver bullet. You see there are unfortunately some deceased patients in both groups, the doctor picking the drug and the machine picking the drug, but there's quite a good response that you get from using machine learning. And so we really want to show that this can actually change lives, and you can use this in a powerful way to learn things about the biology that can have really practical applications. So hopefully what you're saying at this point is, how can I learn how to do this and where can I go for help? Hopefully you're not like, why did I just waste the last hour of my life? The major place to go is forum.image.sc. So this is an open source image analysis forum that combines a ton of tools you've heard about, and probably some you haven't even. We keep adding new tools constantly. This slide is a few months old, and there's probably at least 3 or 4 new partners since then. Where in general, you can upload a picture and say, this is my picture, this is what I care about. Somebody help me figure out what software is the right software to use to learn what I want to know. And you have experts from all of these different tools who monitor this forum, and will go through and they will answer your questions. Some of them are the people who write the software, and some of them are just people who like using the software and are super helpful people, and they go through and they answer questions. So this is always the best place to get information about how to do, in my opinion, any image analysis task you need. You can also look for things like job postings. Our lab is hiring a postdoc right now, so if you thought this was interesting and you want to work with me, we have a postdoctoral training program in image analysis. I would love to see applications. You can find out about cool conferences like AIM. And this is sponsored by what we call the Center for Open Bioimage Analysis, which is a center that is between our group and University of Wisconsin-Madison, the team that runs the ImageJ software, and that we use to engage with the community and make new tutorials, give talks like this, and also to make new tools, for example, deep learning tools and projects to make, for example, ImageJ and CellProfiler easier to use together, which when CellProfiler 4.1 comes out this week, you'll see that there's actually now a CellProfiler module to run ImageJ macros. And with that, I would like to thank all of the people who've done this work and who put this tool together, and the people who paid for it, and you guys for your attention. So we have just a couple minutes left. I can bring in some of the people who've requested to go live and ask their questions.
Oh, perfect. I was just going to see if you did take that note. I'm going to leave now.
I did, yes. All right. Anybody who I've just brought live, if you want to ask a question. Oh, we now have infinitely recurring windows there. Let's go back up here. If anyone has text questions, I'm also happy to take those. Again, unfortunately, we only have a couple minutes since we got a late start, but again, I would also like to thank the organizers. I thanked them at the beginning, but also like to say at the end, thank you guys for putting this together. Yeah, happy to answer questions for as long as there are questions. All right. Otherwise, hope to see you guys all at AIM next year or see you on the forum, and have a good rest of your day. Stay safe and enjoy the rest of the conference. Hey, guys.